All right, thank you all for coming to my talk. I'm Christopher Petty. I'm a biochemistry molecular biology major. I'm also doing my master's in biotechnology. So as a biomedical researcher, what I really endeavor to do is understand the fundamental mechanisms of biological processes. And once we understand these molecular mechanisms, we can exploit them to create novel therapeutics to treat diseases. The process I'm specifically caring about is this process known as myogenesis, which I might also refer to as skeletal muscle differentiation. So myogenesis is the process by which muscle progenitor cells, known as myoblasts, fuse together and form multinucleated myotubes, which are muscle fibers that contribute to all the muscle tissue that you can voluntarily move, which is known as skeletal muscle. And so this process is very critical because not only does this occur during development, it also occurs during muscle regeneration. So understanding the molecular drivers of this process could be very important in making new regenerative therapies to treat muscular diseases. So when trying to understand any biological process, it's always good to go back to the very basics. This is what's known as the central dogma of biology, and it is truly some of the most fundamental aspects of things. So DNA encodes the information of the cell. This is then taken into out into the cell in the form of a copy known as an RNA molecule. And this RNA molecule then dictates which proteins are made. And those proteins do the cellular functions. But we now realize that this process, this paradigm isn't nearly as simple, and there are more complications. Recently, scientists have discovered a novel class of RNA called non-coding RNAs. These are RNAs that do not encode proteins, but instead do the cellular functions directly. And given that there are more non-coding RNAs than protein-coding RNAs, understanding the function of these non-coding RNAs is critical for our understanding of biology. I'm specifically interested in studying what's known as an RNA MegaCluster, known as the Dlk1-Dio3 cluster. So this is a diverse cluster of over 60 non-coding RNAs. They're all produced as one humongous RNA molecule, and then is chopped up into 60 functional units, shown in this complex diagram here. Why we care about this is because people have preliminarily shown that these non-coding RNAs regulate key aspects of skeletal muscle biology, including muscle regeneration, development, and metabolism. But the process of myogenesis has not yet been explored in the context of this non-coding RNA cluster. So in order to study this, the easiest way to get at what they're doing, what these non-coding RNAs are doing, is eliminate them in these muscle progenitor cells, which are shown on the right here, observe if they can form these nice myotubes, and then identify if there are any cellular processes altered in the cells during their formation of myotubes. So in order to eliminate these non-coding RNAs, I'm using a technique known as CRISPR-Cas9 gene editing. So you can think of these as molecular scissors that allow me to cut very precise regions of DNA. So specifically, I'm creating two cuts flanking a particular region of DNA, which is called the promoter, which you can think of as an on-off switch that dictates whether or not these non-coding RNAs are made. And so once I make this deletion, we should expect that there are no more of these non-coding RNAs. Unfortunately, that didn't happen. So in order to observe whether or not I got the successful reduction in these non-coding RNAs, I looked at 3 non-coding RNAs, one at the very beginning of the locus, one at the very middle, one at the very end of the cluster. And if all of those showed a coordinate change in production levels, we would be able to infer the level of the rest of them. And as we see here, this is level of production relative to normal cells, and it's between 1,000 and 10,000-fold increased. So I got the exact opposite result than I wanted. Instead of reducing expression, I've gotten 10,000-fold more than I want. But if I want to know how something works, the second best thing to eliminating it is putting a lot more of it in. So although I do not fully understand how I got this result, it's nonetheless a useful model system for studying these non-coding RNAs. But the question remains, do these non-coding RNAs affect myogenesis? So in order to assess this, I induced the cells to form myotubes and then observed what they looked like. So here we see normal cells. I haven't done anything to them. And I induced them to form myotubes. By this midpoint of the process, we see they begin to elongate and form myotubes, which then become even longer and more robust by the endpoint of this myogenic process. Whereas cells with this increased levels of the non-coding RNAs elongate but don't form mature myotubes. And this this process doesn't get any better by the end of differentiation. To look at that in more detail, I stain the cells for a marker that would be indicative of cells successfully undergoing the myogenic process. So that's shown in green here. But I also stained in blue for nuclei. And so, as I mentioned before, the myogenic process is based off of the fusion of these cells. So we can look at successfully differentiating cells, these successful formation of myotubes, based off of green cells with more than 3 blue spots, which are more than 3 nuclei. And so just qualitatively, again, we can see the impairment of myotube formation. Here we don't see many myotubes. And here we see great myotube formation at the midpoint of the myogenic process. And this is, I can quantify that. And so here we see that normal cells, shown in blue, have there's a 50-50 population of cells that successfully form myotubes versus cells that don't at this midpoint of the myogenic process. Whereas cells with increased non-coding RNAs have substantially fewer successfully formed myotubes, indicating that there is an impairment in this process. In order to look at exactly why this might be, I then looked at whether the cells were expressing, had the correct genes turned on for the myogenic process. And so I'm looking at during this myogenic process, and we see that none of the factors that are known to drive, none of these genes that drive myogenesis are altered at all. And in fact, genes that seem to be, that are markers indicative of successful myotube formation are up, if anything, in the cells with increased non-coding RNAs, indicating that although the cells look like they're not differentiating, at the molecular level they seem fine, which strongly suggests that these cells fail to form myotubes, but due to an indirect secondary process. So there's some other cellular process that's impaired in these cells due to the increased non-coding RNAs. And so that begs the question, what is this process? And so in order to get at all the cellular changes that may have resulted from the increased Dlk1-Dio3 non-coding RNAs, I performed an interesting technique known as RNA Seek. So RNA Seek works by taking all of the RNA in cells, and I did this at the midpoint of the myogenic process for the control cells and the cells with increased Dlk1-Dio3 and non-coding RNAs. So you take all of the RNA and fragment it, and then you sequence it. By sequencing it, you can determine which genes were on and by how much. And by doing this, you can infer all the cellular processes that were active at the time you isolated the RNA. So it's a global molecular snapshot that you can get a lot of information out of. And so by doing this, I got a really rich data set where I found thousands of genes were changed both in the positive and negative direction, which corresponded to, again, thousands of cellular processes. This is just a picture of what my data set looked like. And so clearly I'm not going to walk you through all of those, but I have a couple key findings I want to share with you. So one of the interesting findings was that mitochondria and oxidative metabolism was up. So what does this mean? So mitochondria are the powerhouses of the cell. Thank you. And so the cells with the increased non-coding RNAs are clearly ramping up their energy production for some reason. And this has many interesting implications I can go into in the questions later. But secondarily, I found that the most decreased genes all were related to extracellular matrix. An extracellular matrix is a network of proteins, especially fiber-like proteins, secreted from cells that help the cells adhere to their surroundings, move around, and signal to their surroundings. And so this was a peculiar finding that I wanted to follow up further and just indicate that the findings of this gene set analysis were actually translating into something real that we can observe in the cells. And so to do this, I just assumed that if cells lack something to stick down onto their environment, maybe if you put them in a stickier environment, it will fix things. So that's exactly what I did. I grew the cells in the presence of gelatin to supplement the extracellular matrix that they presumably lack. And control cells, the normal cells, don't differentiate any better or worse, but cells with increased levels of the non-coding RNAs clearly form much better myotubes in the presence of this gelatin, indicating that this increased adhesive substrate really helps their differentiation. So the key findings of my project so far have been to establish a novel cell line that I can study these Dlk1-Dio3 non-coding RNAs in, because they have dramatically increased levels of these non-coding RNAs. The altered dosage of these non-coding RNAs impairs the myogenesis process, although likely through an indirect mechanism. And I've identified many cellular processes that are altered in these cells with increased Dlk1-Dio3 non-coding RNAs. So that was a lot of details. Why do we care about all this? Let's get back to the big picture. Understanding the molecular mechanisms of the Dlk1-Dio3 non-coding RNAs can and already is translating into non-coding RNA-based therapeutics to treat muscular diseases and can serve as drug targets similarly to treat muscular diseases. Thank you.
So you mentioned that the raised level or number of mitochondria had several interesting implications that you couldn't go into during the presentation. Can you go into them now?
Well, an interesting aspect of this entire project is it seems like everything that I did went the wrong way that we would expect. And that's true of this. There's reason to believe that these non-coding RNAs should be negative regulators of mitochondria. So it's fairly controversial that I'm finding that they're increased. But furthermore, outside of that, just simply based off of the understanding that mitochondria are the powerhouses of the cell, they're very important in this metabolic process. It would support the notion that the cells are differentiating, forming these myotubes even better, because we expect that myotubes need to have a lot of energy. And so that's indicative that they have some processes that may be even more muscle-like than they should be, which is interesting.
What kind of diseases are we talking about?
So in terms of what I might fix? Or
potential therapeutics like dystrophy.
Interestingly, so muscular dystrophy is the easy one to hit, but the interesting thing is that it's being used as a target for sarcopenia and muscle aging as well. Overexpression of some of these non-coding RNAs is sufficient to reverse sarcopenia, which is very interesting. Sarcopenia is a wasting of muscle in old age. All right.